Hello. So we have stopped somewhere here itself in the same slide. Let me continue with that. So heteropolysaccharides are the polysaccharides that are formed of more than two or more than two types of monosaccharide units. Okay. See, I'll just draw and so that you will be familiar with that. Yes. So here, in case of homopolysaccharide, they are formed of linear chain of any monosaccharide unit. Heteropolysaccharide is also a chain of monosaccharide unit, but the difference is that here the repetitive units are different. Okay, it takes time anyway, but you'll have to stay with me for this. So this is what it is. Okay, I think I can draw it on this side also so that it will be better. Okay, clear. Now what you see here, it is formed of single type of monosaccharide unit, whereas here it is formed of more than one type, two types of monosaccharide unit, or it might be more than that. The chain continues anyway. Either you can take this or you can consider this as one unit. Okay, it gets repeated. Okay, so heteropolysaccharides are the polysaccharides formed of more than one type of monosaccharide unit, and they are present in they provide extracellular support to organisms of all different kingdoms. Okay, and example I am repeating again in this class: hyaluronic acid, heparin, chondroitin sulfate, and keratin sulfate. Okay, and now let's see one by one these heteropolysaccharides. They are generally known as glycosaminoglycan. Example is hyaluronic acid. Now this is a polymer of two units, that is, one is D-glucouronic acid, the other one is N-acetyl glucosamine. What you are seeing in the picture are, one is D-glucouronic acid, the other one is N-acetyl glucosamine. Let me use a pen here so that I can focus it. So what you are seeing here is this is the glucouronic acid. Okay, and we have N-acetyl glucosamine. N-acetyl glucosamine here, acetyl group is attached anyway, you can see here N-acetyl glucosamine is present. So these two are the monosaccharide units that actually form disaccharide now, because they are going to repeat in bundle as this is going to be repeated in throughout the length of the hyaluronic acid. And how many such units are found? Around 50,000 such repetitions are found. So in homopolysaccharide it will be only glucose, but in heteropolysaccharide more than two types of repeating units are found. So here one is this glucouronic acid, the other one is N-acetyl glucosamine. So this unit is going to repeat. How many times? Up to 50,000 repeats are present, okay, in hyaluronic acid. And this hyaluronic acid, it gets its name because in Greek hyalos means glass. Okay, so hyaluronic acid, it will be having a glassy and or a translucent appearance. That's why this particular carbohydrate is called as hyaluronic acid. Now where do you find them? You find them in the vitreous humor of vertebrate eye. So, and it will have a jelly like consistency. So you can see here. Okay, so this one, this is the hyaluronic acid composition that gives the jelly like consistency to the vitreous humor. You know, eye has aqueous humor and vitreous humor. Vitreous humor actually gives the shape to the eyeball. So that is the hyaluronic acid that is present. Not only that, it is also present in the synovial fluid of a joint. So here you can see, so synovial fluid is present. Okay, this synovial fluid is also formed of hyaluronic acid. Remember, hyaluronic acid gets its name because of the glass-like appearance or translucent appearance. In addition to that, it is also present in the extracellular matrix of the cartilage and tendons. Okay, tendons that connects the muscles to the bone. Then it is also present in almost all different tissue matrix. You take a areolar connective tissue matrix or this one adipose tissue matrix. So the matrix of these connective tissues is basically formed of hyaluronic acid, which gives, and the composition or the amount of this hyaluronic acid might vary depending upon the type of tissue you see. Okay, and it is also present in between the cells. And now here we are talking something what you might have already studied in your previous semester, hyaluronidase. This is an enzyme that will be that will be secreted at the time of fertilization, that will help in separating the connection between different cells around the ova. Okay, and what is that composition that binds these different cells together? It's a hyaluronic acid. So this hyaluronic acid will be separated by the enzyme hyaluronidase, and so this actually helps in separating it out, and this is present even secreted by some pathogenic bacteria too, so that they can break the tissue and penetrate. How bacteria is able to penetrate through all the primary defense systems, barriers of our body, is because they are provided with different enzymes. Hyaluronidase is one such kind of enzyme, okay, that can penetrate the cells or the tissues and then can infect us. And this hyaluronic acid, its significance is that it is the major composition of matrix. Okay, and it is very highly difficult to imagine a matrix without the hyaluronic acid. So remember, it is a polysaccharide. It's a heteropolysaccharide formed of glucouronic acid and N-acetyl glucosamine. The next type of polysaccharide we will be discussing today is heparin. Heparin, so it gets its name from Greek. Hepar means liver. Okay, and this is a natural anticoagulant. Okay, heparin is a natural anticoagulant. That means it prevents coagulation of the blood, and it is made in mast cells. They are separate special type of some white blood cells present in our body, and it is a sulfur rich glycosaminoglycan. Glycan means a polysaccharide, you know. So a glycosaminoglycan is a it's a sulfur containing compound, as you can see here. So sulfur containing compound it is, so rich sulfur. Okay, and it is formed of disaccharide, repetitions of disaccharide units, and the repeating disaccharide units are sulfated ioduronic acid and sulfated glucosamine. Both together they form the repeating units of heparin. This heparin, it is a anticoagulant, we said, and it prevents coagulation of the blood by binding to a protein called as antithrombin. So this antithrombin will then bind to thrombin. When heparin binds to antithrombin, antithrombin will bind to thrombin, so that no activation takes place and no blood coagulation occurs, clotting occurs. And it has a high it is used in preventing clotting. Okay, when you have to store blood for longer time, this heparin can be used. Of course, there are other methods of like some chelating ligands are also used. Citrates and all are used as anticoagulant, but heparin is one such chemical or a polysaccharide that can be used in preventing the clotting of blood when you are storing the blood for a longer time. Okay, and there is one general information. This is the highest negative charge density. It has the highest negative charge density among all the biological macromolecules that are known to human. Okay. The next type of heteropolysaccharide we are studying is chondroitin sulfate. So chondroitin sulfate is again from Greek. Chondros means a cartilage. So chondroitin sulfate is one of the component of your cartilage. You know, cartilage has its soft nature, slightly elastic nature also, and it is its matrix is formed of chondroitin sulfate, different from the bone. Okay, bone is generally formed of inorganic minerals, whereas chondroitin sulfate forms the matrix of cartilage. So because it forms the matrix, it is called as chondroitin sulfate. Sulfate because it contains sulfur group, anyway. Chondroitin sulfate because it is a composition of cartilage or a matrix of cartilage. This chondroitin sulfate, it is a sulfated glycosaminoglycan, again one more glyconated compound, and its disaccharide units are N-acetyl galactosamine and glucouronic acid. I am going to the previous slide here. So it is a sulfated iodouronic acid and sulfated glucosamine. Okay, that is glucosamine, whereas chondroitin sulfate is formed of galactosamine, acetyl galactosamine. Remember, glucosamine and galactosamine are different because glucose and galactose differ in one of their OH positions. Okay, usually in the fourth carbon atom, OH group is above and below the chain in case of glucose and galactose. Okay, so this is formed of N-acetyl galactosamine and glucouronic acid. Heparin is formed of iodouronic acid. Okay, so this is a GAG. GAG means glycosaminoglycan. Now this contributes, I said it's present in the cartilage, it is also seen in tendons and ligaments, and remember wall of the aorta too. Aorta, the largest vessel that carries the blood to different parts of the body away from heart. So this, its wall is also formed of chondroitin sulfate, providing some kind of elasticity as well as high tensile strength. Okay, so this is used in as a dietary supplement to treat osteoarthritis also. So arthritis, joint inflammations are seen generally in aged people, but sometimes even there are certain immune disorders too where arthritis might appear. So in case of osteoarthritis, joint inflammations, this chondroitin sulfate is used as a dietary supplement. Okay, now let's just summarize the different functions of carbohydrates. So they are the most abundant dietary source of energy. So 4 kilocalories of energy is generated by a gram of carbohydrate, and it is glucose is broken down during glycolysis and Krebs cycle to give ATP during oxidative phosphorylation, and it also stores the energy in the form of glycogen in animals and starch in plants. So it also forms a structural element. So the murin in bacteria, cellulose in plants and animals, chitin. Arthropoda, their body is basically formed of, exoskeleton is formed of chitin. Chitin is nothing but a homopolysaccharide formed of N-acetylglucosamine. N-acetylglucosamine residues form the repetitions in chitin. Cellulose is a polysaccharide of glucose. Okay, so they are the rich source of energy, they are the storage structures as well as they form the structural elements. They are also associated with the different lipids and the proteins, and in that case they form surface antigens, receptor molecules, different vitamins, okay, even antibiotics. Not only that, the carbohydrates are also components of structural framework of RNA and DNA. Remember, ribose and deoxyribose. Okay, and they also help in cell-cell communication and interaction between cells. There are a number of such functions, uses, biological significances of carbohydrates. And these are my references for the whole class, or almost all the different concepts of carbohydrates. I have referred to these websites as well as Ranger's book. Okay, and most of the pictures are also from these. Thank you.